Okay. Okay. Everyone, please confirm me. Am I audible to everyone? Everyone, please confirm me. Am I audible to everyone? Good morning, everyone. Please confirm. Am I audible to everyone? All right. Good morning, everyone. Good morning. Okay, so today I'm going to solve, the TSSET question paper. I'm requesting all the CSR net people also, actively solve all the questions, without fail. there are total there are total 100 questions are there. Okay, now remember, every one of you, I want you people, at least, I want you to solve 70 to 75 questions very correctly. Okay, especially if the CSR net people can answer 70 to 75 questions very correctly, trust me, your preparation is also going in a right direction. All these questions can help us in the part B level. Okay, and the people who written this particular TSSET exam, there are many number of questions that you can object. There are more than 7 questions you can object. Okay, and if you object this particular 7 questions, the people who are very near to the cutoff, you are having 100% of chances are there that you can easily qualify the exam. And I will tell you very clearly what kind of questions that you can object. I'll give you valid explanation with the references. You can use the same references and valid explanations, to object your question. All right. Okay, now everyone, please be patient. The session will go beyond 2 hours. Please be patient and please involve and try to solve every single question without fail. All right, now let's start the question number 1, or the 51. Everyone, think properly. I want every student, remember my words, out of 100 questions, I want everyone to answer 70 to 75. And please tell me how many number of questions you are doing correctly at the end. All right. Question number 1. Which of the following statements are correct? Polarity of water makes it an excellent solvent. Water has the highest tensile strength. Cohesive property of water, as well as water has high dielectric constant. No, I mean, now my side internet connection is absolutely fine. You just refresh your side. Okay. Okay. There are answers were given in the question, but most of the answers are very wrong. Okay. Most of the answers are wrong. I want you to give me the answers. Everyone, please give me the answer. Saumya, Shaila, Padmavati, Shivani, try the answers. Bhavani, which is a basic question. I want every one of you to give the answers little faster, friends. By that we can go, little bit fast. Everyone, which of the following statements are correct? Okay, now option 1 says that polarity of water makes it an excellent solvent. Of course, we know very well water can act as a very good solvent. It can dissolve most of the substances. Chandar, please look into the options very correctly. The next one, water has the high tensile strength. This statement, I can say that 60 to 70%, this statement will be the correct. But compared to the all options, we can say that this option is also true option. What is called tensile strength? Tensile strength means the ability or capability of a molecule to tolerate under the stress conditions. Understand once again, the ability or capability of the molecule to tolerate under the stress condition. That is what we generally call it as the high tensile strength. Now you know, right, why water molecules will be having the high tensile strength? Because they are having the hydrogen bonds. Hydrogen bonds are the one which will provide the tensile strength. So the option 2 is also right option. Then cohesive properties of water. We read in the xylem also, right? How one water molecule can be combined with the another water molecule. We read it. So one water molecule can be associated with one other water molecule with the help of a cohesion. Undoubtedly option 3 is also correct. If you just come to know that the option 3 is correct, you can easily eliminate options. Now water has the high dielectric constant, which is a common statement which we read in the 11th standard also. Option 4 is the correct answer. Okay, I kept every question with a very valid explanation, which you can use this explanation to object the question, right? So as I said, water having the high polarity, because water having what? Partial positive charge, correct na? And it is also having the partial negative charge also. Because of the partial positive and partial negative charge, the water molecule can act as an excellent solvent. That is very correct. Then I said water has the high tensile strength. Why water is getting the high tensile strength? Because of the presence of the strong hydrogen bonds. Option 2 also absolutely true. Cohesive properties, I have very clearly told you. Cohesive property means joining one water molecule with the another water molecule exclusively with the help of a hydrogen bonding. Absolutely true statement. And what is called as a dielectric constant? It is the ability to reduce the electrostatic forces between the two charges. Undoubtedly friends, water having the high dielectric constant. All the 4 statements are correct, hence the option 4 is the true option. No point of getting confused. I have kept a very crystal clear explanation. I want every one of you to understand that correctly. All right. Now every single student, please answer the next question. The peptide bond in a protein is. Most of the answers are wrong. I want you to give me the answer with a valid explanation. The peptide bond in a protein is. Friends, please don't take too much of time. Please try to answer the question in 20 seconds to 30 seconds. It is very easy questions. By that it will become, good practice to all of us. And I kept the explanation why the other options are wrong. Everyone, what is the peptide bond in a protein is? They are simply asking, friends, the characteristic of a peptide bond. Everyone, Vijay Lakshmi, what is the answer? Shivani, Bhavani, what is the answer? Come on, why you are people delaying the answers? You know, right, friends? First of all, where the peptide bond will be formed. I believe everyone remember. Why you people are not answering such a simple questions? We have a central carbon. We have a right side carboxyl group. We have a left side amino group. Correct na? And we have one hydrogen and we have the one R group. Correct na? Then again, if you look into the another amino acid also, carboxyl group, hydrogen, R group, then followed by the amino group. And you all people know very well where this particular peptide bond can be formed. Peptide bond can be formed by the mechanism called as what? Condensation, that is loss of water. So the peptide bond is generally formed between the CO and NH group. Correct na? So what is this? This bond is located on a single plane. This bond is located on a single plane. So basically it is what? Planar. There is no doubt about it. It is planar, and most importantly, the carbons associated with bond are always arranged in a opposite direction. That's the reason we call it as the trans configuration. All the students, please look into the picture correctly. What are the various characteristics of the peptide bond? It will show the planarity. What is called planarity? The bond between the carboxyl group and amino group. This is called as the planar bond. What happens because of the planar bond? Planar bond is a kind of a covalent bond which is extremely rigid. And we all know very well all the time this peptide bond will be in the trans configuration. Every student generally know that what is called trans configuration, where two alpha carbon atoms attached to the amino acids are on the opposite direction. You can see this, friends. This is one carbon atom and this is the another carbon atom. So both carbon atoms are diagonal or they are opposite to each other. That is what we can call the trans configuration. Then obviously the answer will be what? Option 3. It is a planar and found in the trans configuration. One of the most important statement every student has to remember, why it exists in the trans configuration. That is most important here. It exists in the trans configuration to avoid the steric hindrance. To avoid this steric hindrance, all the time the peptide bonds are arranged in a trans configuration. Option 3 will be the absolutely true option. No need to get confused with this particular point. Every question is just directly from the slides itself. You can answer it very clearly. Now, all the students answer the next question. 54. Amino acid tend to be amino acid tend to be least soluble in water at their isoelectric point. And the reason, the net charge on amino acid is a function of the pH of the solution. Swati, tell me what is the answer for this question? Everyone think properly and answer the given question. It's a straightforward question which is there in the PPT which we already read it. Amino acid tend to be least soluble in water at their isoelectric point. Of course, I have very clearly told to all of you one common point. What is that common point? Everyone, anyone can answer the question. If you look into this picture, what generally amino acid having? Amino acid having carboxyl group, amino acid having the amino group. Correct na? And there is a hydrogen and there is a R group. Correct or not? Now, what happened? When I dissolve the amino acid in the water, I believe every student remember what happened. This carboxyl group lose the hydrogen and it is converted into the COO minus, which is the acidic. And what about this hydrogen? This hydrogen can be accepted by what? NH2. NH2 and NH3 converted into what? NH3 plus. So which is called as basic. So amino acid having the basic also, amino acid exists in the acidic also. Now what do we call this kind of amino acid? We read 4 or 5 times. We call it as a Zwitter ion. Correct na? What do we call it as the Zwitter ion? The net charge, now what is called as actually the Zwitter ion? The amino acid where it will possess the acidic also, the amino acid where it will possess the basic charge also. That is what exactly we call it as what? Zwitter ion. No point of getting confused. So undoubtedly the assertion is true. There is no doubt about it. The assertion is true. And what is the reason they have given? The reason they have given is the net charge on an amino acid is a function of the pH of the solution. Now friends, tell me, what is the relationship between, every student answer my question. Every student answer my question. What is the relationship between net charge and pH of the solution? Everyone answer, is there any relationship between the net charge and pH? Everyone. Everyone. Obviously, there will be no relationship between the net charge and pH. Correct na? So remember everyone very correctly. The net charge on amino acid does not depend on the pH of the surrounding solution. I believe we have studied it very clearly, friends. Depending on the pH, I believe you remember, at the acidic pH, amino acid can act as a positively charged, correct na? Or negatively charged at the basic pH, correct na? At the neutral pH, amino acid can act like a Zwitter ion, correct or not? So net charge is not at all dependent on the pH. Hence, friends, this particular reason is not correct for the assertion, correct na? But reason and assertion are true, but reason is the not correct explanation of the assertion. Option 2 will be the correct option. No point of getting confused over there. Everyone, next question number 55. I want every student has to answer this question very correctly. Okay, think properly and answer. Please be patient, we have to solve many questions. The question says that which of the following statements about bDNA structure is incorrect? They are asking incorrect, friends. It's a very straightforward question. It is also 11th standard question only. Okay. Everyone just give me one moment of time. You please see that. Okay. Yeah. So I hope every one of you understand the point very correctly. I believe everyone remember, right? If you look into the structure of the bDNA, friends, what we discussed, one turn, what we can call one turn of the bDNA we directly studied in the slide itself. It contain 10 to 10.4 base pairs. There is no point of getting confused over there. Next one, the double helix contain what? Anti-parallel chains, because we know very well one strand will be moving in the 5' to 3' direction and another strand will be moving in the 3' to 5' direction. There is no doubt about it. But so option 2 is also correct option. Everyone please look into the option 3. The amount of adenine plus thymine is equal to the amount of the guanine plus cytosine. That statement is absolutely wrong. What is according to the Chargaff's rule? According to the Chargaff's rule, I hope every one of you remember that. What is according to the Chargaff's rule? The number of adenines is equal to the thymine and number of guanines is equal to the cytosine. Correct na? There is no rule that says that A plus T equal to G plus C, which is absolutely wrong statement. Correct na? So option 3 will be the incorrect statement. Friends, don't get confused. They have not asked correct statement. They have asked incorrect statement. They said that A plus T equal to G plus C. That is wrong statement. We can say the A equal to T plus G equal to C. No point of getting confused over there. Next, option D says that separation of the two strands of the double helix require untwisting of the helix. Absolutely true statement. Hence, option 3 will be the incorrect statement. Very straightforward question only. Okay. Everyone please answer the question. This is actually the question which is asked in the CSIR net exam also. I think in the 2022 paper, they asked the same question. Now they asked in the TSSET also in the part B. Swati, what is the answer for this? Glucose and mannose are epimers. What does it mean? Glucose and mannose are epimers. We already read the structures very clearly. Okay. Vijay Lakshmi, Shivani, every student tell me very clearly. Glucose and mannoses both are aldoses or ketoses? Padmavati, glucose and mannoses, are they aldoses or ketoses? Everyone has to answer the question quickly. Glucose and mannose, are they aldoses or ketoses? Padmavati? Obviously, glucose and mannoses are what? They are aldoses. Correct na? Every one of you pay attention towards the structure very correctly. If you observe the structure of the glucose, at the terminal position it is having aldehyde group. At terminal position, even mannose also having what group? Aldehyde group only. There's no doubt about it. Then what is the only difference? The only difference is that if you look into the glucose, on the second carbon, OH group is located on the right hand direction. If you look into the mannose, OH group is located on the left hand direction. Correct na? So there is only one difference. Epimers are the type of diastereomers that differ in the configuration around one specific carbon atom only. There is no point of getting confused over there. It is very easy to understand. Hence the correct answer is option 4. See, I kept the explanation very clearly because there are many questions that you can object. I will tell you the number of questions to object. Kindly use this explanation and you can object the question. Then only you can get the additional marks which help you to crack the exam. All right. Very straightforward question. Swati, try this also, everyone. This is directly from the PPT. Akila, which of the following statements are correct? Now I want you to tell me why they are correct. Option 1 says that double bonds in unsaturated fatty acids are generally in the cis configuration. In polyunsaturated fatty acids, double bonds are almost never conjugated. Sphingoglycolipids are involved in biological recognition. Prostaglandin contain 5-membered ring structure. Prostaglandin regulate the synthesis of, everyone try to understand, intracellular messenger cyclic AMP. It's not chemistry paper, it is a life science paper. Okay, it's not chemistry paper. Life sciences contain biochemistry, right? So these are all the questions from the chapter of the biochemistry. Okay, it's not chemistry, it's a biochemistry. Got it? All right, all right. Everyone, please try to eliminate the wrong option. You can come to a conclusion. Okay. Now, tell me, every student, during the time of cell biology also I told you, unit 1, unit 2, unit 4 also we read. Now tell me, Selvi, do you heard anywhere that the prostaglandins are involved in the regulation of the cyclic AMP? Every student, please kindly answer. Do you read anywhere that the prostaglandins are involved in the regulation of the cyclic AMP? Obviously not. We all studied very clearly adenylyl cyclase is the effector molecule. It is the one which is converting the ATP to the cyclic AMP. Correct or not? And when this particular adenylyl cyclase will be acting whenever a hormone or a specific growth factor comes into the picture, then it will come. Hence, you can easily eliminate the option 5. Option 5 is absolutely wrong option. Hence, you can eliminate the option 5 easily. Now, how many number of times we have studied the same line in the slides in the PPT? Double bonds in unsaturated fatty acids are generally in the cis configuration. Double bonds, we already studied, friends. We have a MUFA, monounsaturated fatty acids, and we are having another one. We generally call it as what? PUFA, polyunsaturated fatty acid. In case of a MUFA, there is only one double bond. In case of a PUFA, there is more than one double bond. Sometimes it may be a double bond, or sometimes it may be a triple bond also. I believe every student remember whether it is a monounsaturated fatty acid or polyunsaturated fatty acid. All the unsaturated fatty acid double bonds are always in the cis configuration. No doubt about it. Correct na? And in PUFA, double bonds are almost never conjugated. They never combine with other. Correct or not? I have very clearly told you, friends, double bonds, they are having low melting point. Correct or not? Always They are having the low melting point. Best example, you can take all our vegetable oils. Why? What is the reason? They never get conjugated. So melting point is also low. We can melt very easily. Next, sphingoglycolipids are involved in the biological recognition. The best example, you can take an example of sphingosine, which are involved in the formation of the myelin sheath, which is obviously involved in the biological recognition. And I hope you all people remember my statements. Prostaglandins are the major chemical mediators which are involved in the mechanism of the inflammation. Prostaglandins are basically what? They are the ringed structure. If you look into the normal fatty acid, they are straight chain. But if you look into the cholesterol and prostaglandins, they are basically ringed structure. Correct na? The ring named as cyclopentanoperhydrophenanthrene ring. Correct na? Option 4 also correct. Hence the correct answer will be option 1, option 2, option 3, option 4. Right. I want all the CSR people also has to give the right answer. These questions are very small level questions for all of you. I want every student has to give the correct answers. I kept the answers with a crystal clear explanations. I want every one of you to go through it. All right. And this is the straightforward question which we read 4 or 5 times in the explanation itself. Tell me, Swati, what exactly Michaels-Menten equation states that? Bhavani, Shivani, what exactly the function of a Michael-Menten equation? Correct. What is the function of a Michaels-Menten equation? What it is saying us? I hope every student remember what is MM. At what concentration of the substrate? Every student understand. At what concentration of the substrate the enzyme reaches half of its Vmax? Correct or not? I explained 4 or 5 times the same thing. At what concentration of a substrate the enzymes are releasing half of its Vmax? Correct na? Very good. Rate of reaction in terms of the substrate. In other words, the concentration of the substrate where the enzyme is reaching half of its maximum velocity. Every student forgot it. Na? Remember, what is equation? 1 second. Velocity V equal to Vmax substrate by Km plus substrate. Correct or not? What is Km? Km is the Michaels-Menten constant. Everyone, correct or not? What is the Michaels-Menten constant is actually explaining us. Michaels-Menten constant is explaining us at what substrate concentration the enzyme reaches half of its maximum velocity. Very good. And this question also you can challenge. In the given answer key, they have said that Km is the enzyme substrate complex association constant. Why we can challenge this question is when we are using the word Km. Every student listen. Why you can challenge? When you are using the word Km, you cannot use association constant. You cannot use the association constant because association constant can be represented by a Ka and dissociation constant can be represented by the Kd. Association constant Ka, association constant Kd. But in the given question, they says that Km is the enzyme substrate complex association constant. That is absolutely wrong statement. And if you look into the option 4, enzyme substrate complex formation is essential for enzyme reaction. This statement is true, but that is not at all explained by the Michaels-Menten equation. Hence, you people can object this question. I kept a crystal clear explanation of the Leninger and White book. you can raise the objection, there is a great possibility that they can give the marks. You never say that Km is the association constant. Km only explain the at what substrate concentration enzyme reaches half of its velocity. That's all. This is only the true option. Hence, you can object the question number 58. Okay, don't forget. Okay, all right. I want every student has to answer this question. Everyone has to give the right answer. This is also directly there in the slide itself. The first and second slides of the transposable concept that what we are discussed, it is there. It slowly answer the question and give me the correct conclusion. I want everyone to give the correct answer. Slowly think and answer the given question. Eliminate the options crystal clearly. There is no point of getting confused over there. Eliminate the options. Tell me what is the why everyone. All right. Which of the following statements are correct about retrotransposons? We all people studied multiple times. There are two type of a transposable elements are there. One is called DNA transposable elements. Another one is called as the RNA transposable elements. RNA transposable elements, what do we also call it as? Correct. We also call it as the retrotransposons. We all studied crystal clearly the retrotransposons always can be, undergo the transposition by using RNA as their intermediate. Correct or not? So option 1 is absolutely true. They transposes via RNA intermediates. Every student, please look into this picture very correctly. See, this is the retrotransposon. I hope everyone can see the picture very clearly. This is the retrotransposon. They can undergo the transposition mechanism by using only RNA as their intermediate. Absolutely true. Now tell me everyone, do you really think retrotransposon, does it code for RNA polymerase enzyme? Does retrotransposon code for RNA polymerase enzyme? Never ever. Do you think retrotransposon can it code for the ribosomes? Never ever. It never code for the RNA polymerase. It never code for the ribosomes also. Correct na? Hence, option 2 is wrong because they said that they contain genes found in ribosomes and they can code for RNA polymerase. That is also wrong statement. They are found only in bacteria. This statement is absolutely wrong. Retrotransposons widely found in the eukaryotes. I have crystal clearly told you the same statement 4 or 5 times when I am teaching you the lesson. I hope you remember. Bacteria basically contain what? DNA transposons. They may contain the introns. Obviously friends, every gene sequence generally contain the introns. So option 5 is also correct. So only option 1 and option 5 are the correct options. Remaining all options are the wrong options. There is no point of getting confused over there. I kept the very elaborative explanation. I want every one of you to go through it. All right. See, such a simple question. This is also a question of 11th standard. Everyone, please answer the given question. Everyone, which of the following accurately describes the sodium potassium ATPase pump's role in the cellular physiology? Which of the following correctly describes or accurately describes the function of sodium potassium ATPase pumps? So easily you can come to the conclusion. You no need to look at the options also. Okay, I'll write the option 4 here. Option 4 is 2 and 3. Undoubtedly, we know that sodium, potassium, ATPase pumps. So, sodium, potassium, ATPase pumps, we know that they never ever involved in the passive diffusion, or they never involved in the facilitated diffusion also. And we know that in order to pump in order to pump sodium and potassium, it is strictly dependent on what? It is strictly dependent on the ATP. Correct or not? So undoubtedly option 2 will be the option 2 will be the very accurate option. And option 3 says that it regulates the water movement. Why the sodium potassium ATPase pump regulate the water movement? Water movement can be mediated by the aquaporins or the water channels. And option 4 says that it is operate independently of the energy. That statement is wrong. It is strictly dependent on energy. Hence, the only correct option is the option 2. Very good, very good, very good. Correct, correct. Okay. You know, every student take lot of time and answer this question. Okay. I am not giving you the options also, but I want you to answer the question. At least try it. At least try it. I don't, I don't worry whether you are answering every question or not, but make sure that you answer few questions correctly, at least 70 questions. Take your time. I am not showing the options also. We read it. I have explained you every single word in the microbial physiology. In the microbial physiology, there is a specific slide where all these words are written. Every word. In the unit 2. Swati, recall and answer the question. Akila, Shivani, recall this. Take your time, no problem. Simple it is. If you use a little bit common sense also, you can answer this question. Swati, try it correctly. It is easy question. We can do this. Wonderful, Padmavati. Very, very, very good. Very good. Wonderful, wonderful. Very good. Very good. See, very simple question it is. Option A says that activation of heat shock proteins to repair damaged DNA. First of all, what is the major function of the heat shock proteins? Heat shock proteins are major type of proteins which are involved in the protein folding. Correct na? At the same time, heat shock proteins will be, released during the time of a DNA damage. And what this heat shock proteins will do? Heat shock proteins will stimulate the repair mechanisms. So option A, option 1. Upregulation of antioxidant enzymes. I have told you multiple times, whenever you heard the name of a antioxidant, what is the common statement you have to remember? Antioxidants are the one which are going to destruct free radical formation. Correct na? What are free radicals? You remember, reactive oxygen species are free radicals. You can take an example of H2O2, you can take O2 minus. These are all what? Free radicals. So how can we destruct this particular free radicals? We can destruct any kind of a free radicals by using what? Antioxidant enzymes. Obviously B will be the 2. Option C, inhibition of the protein synthesis to conserve energy. Correct na? Now you tell me, if I want to inhibit the protein synthesis, tell me what happened. If I inhibit the protein synthesis, obviously what happened? Metabolic activity will be reduced, because entire metabolism, You consider the catabolism, you consider the anabolism. Everything is exclusively depends on what? Everything is depends on the proteins only, no doubt about it. So if you inhibit the synthesis of the protein, obviously there is a reduction in the metabolism. Obviously C will be the 3. Now, increased production of osmoprotectants to maintain the cell membrane integrity. I believe, can any student give me the example? You know, the same question they have asked in the same question paper in other way. I will show you. Best example, you can take osmoprotectants such as proline. You can take betaine, and you can take a lot of sugars such as mannitol. This function is what? Very good. What is their function? Help the cell to tolerate to the environmental stress condition, especially friends, heat stress. During the heat stress, all these osmoprotectants will be released generally. Wonderful. Then D will be the 4. Very good. Whomever given the right answers. Wonderful. Very good. Proline, betaine, mannitol, all these are involved, are acting like osmoprotectants. This is also straightforward slide. Very good. Very good. I kept a very elaborative explanation. Please go ahead. Okay. This is also a question of 11th standard. This is the question of 11th standard. Okay. Look into the given question. What is the question? Says that everyone please look into the question. Peroxisome play a crucial role in the detoxification process, particularly the breakdown of hydrogen peroxide. This is the first line in the PPT, in the peroxisome organelle. Then the main function is store calcium and regulate intracellular calcium levels. Anyway, can any student quickly give me the answer? What is the name of the cell organelle? What is the name of the cell organelle that store the calcium and that regulate the intracellular calcium levels? Every student answer. What is the name of the cell organelle that store the calcium and that regulate the intracellular calcium levels? What is that name of the cell organelle? Very good. So always remember friends, SCR is the one which will store it. Correct na? And sometimes they will say that mitochondria also involved in the regulation. I am not saying that storage, okay? Mitochondria is involved in the regulation of the calcium levels, but both storage and regulation can be mediated by this particular SCR only. Anyway, the option 3 is the correct. The assertion is correct, but the reason is incorrect. Very good. And this is the question every student, I am telling you right now, immediately object to this question. Immediately. Okay. Undoubtedly, you will get the marks. Okay. Okay. Now tell me Swati, tell me Selvi, what is the function of the APC? I want every student has to answer. APC. Now, tell me Swati very correctly, does the APC having any role in the S phase? Akila, Selvi, my question is Shivani, does the APC having any role in the S phase? Everyone. Vijay Lakshmi, slowly, don't be hurry, okay? Does the APC having any role in the S phase? Tell me Swati very clearly where we use the APC. Akila, where we use the APC? It is anaphase promoting complex. It is only responsible for allowing the cell to enter from metaphase to the anaphase. It is only involved in allowing the cell from metaphase to the anaphase. Correct or not? That's the reason we call it as the anaphase promoting complex. It do not have a direct or indirect role in the S phase. Then option 2, APC degrades cyclins and other regulatory proteins to allow the progression through the cell cycle. I will tell you one statement. This statement is partially correct. Why? Because I will tell you. I strongly believe every student remember what is the function of APC. APC will allow the cell to enter from metaphase to anaphase. If it wants to enter into from metaphase to anaphase, it wants to degrade some proteins. It will degrade securins. It will degrade cyclins. Correct or not? It will degrade only in the M phase. Okay, only in the M phase. It do not have any role to allow the progression after the DNA replication. This statement itself wrong statement. Okay, I can consider allow progression is a part of the right answer, but it never ever involved in the DNA replication. Never ever. So every student, please look into the crystal clear explanation. I want every one of you understand the given explanation. Statement 1 says that APC initiates during the S phase. The S phase is the phase where the DNA replication occur. Correct or not? However, APC is not active during the S phase. APC do not have any role and it is not active during the S phase. Then how it is involved in the DNA replication? It is absolutely wrong statement. Then APC degrade the cyclins and other regulatory proteins throughout the cell cycle. I believe everyone remember APC only degrade securin and it will degrade cyclin B. Correct na? But cyclin B and securin, where they are present? They are present at the anaphase. So only in the metaphase to anaphase only the APC function will be there. So option 1 is completely wrong option. Okay, this question you can immediately object. Okay, trust my words, if you object, 4 marks are in your hand. No doubt about it. Okay, this completely wrong. All right, very straightforward. Again 11th standard question. See the level of the set standard, very small level it is. Everyone, what is the function of an operator in an operon system? Please be patient. I want every one of you to present till the end of the class. What is the function of an operator in an operon system? Anyway, you all know that what is the function of an operator? Operator is the one which is acting like a switch. Correct na? Everyone, why you are thinking this much of time? Correct na? How many times we read the question? 10 times we read it, everything. So you know that promoter is the binding site where RNA polymerase will come and attaches. Operator is the binding site where the repressor will come and attaches. If operator got blocked, RNA polymerase enzyme cannot cross the operator. If it is not crossing the operator, then there will be no gene expression at all. Okay, see, such a simple question they have asked in the exam, which is just 11th standard question. All right, this is also exactly 11th standard question. Exactly 11th standard. During which stage of the prophase 1 in meiosis does synapses occur leading to the formation of tetrads? Very simple. During which stage of the prophase 1 in meiosis does synapses occur leading to the formation of a tetrads? In simple words, in which stage crossing over takes place? We all generally know, right? Zygote. I hope you all people remember this particular statement, right? Everyone. Zygote is the state where the synapses starts between the non-sister chromatids. Correct na? Homologous chromosomes pair up closely to form the tetrads, which we generally call it as what? Bivalents. No point of confusing at all. Simple, straightforward question. Okay. See this there are two questions asked only from the transposable element. One question they have asked about the RNA transposons. One question they have asked straightforward about how transposons are typically arranged in the genome. We read the same lines in the PPT. How transposons are typically arranged in the genome. Everyone. See, of course, friends, we all studied, right? What does the transposable enzyme contain? Suppose this is the transposable element. What else it contain? Transposable element contain terminal inverted repeats. Correct or not? Terminal inverted repeats. And what do we call them? We call them as the flanking direct repeats. Correct or not? And how the transposable elements are arranged? See, suppose this is the gene. Here also transposable element is present. Here also transposable element is present. At many locations, the transposable element is present. So what is the meaning of that? They will be scattered. They will be scattered or arranged throughout the genome. No point of confusion. This is the direct statement, which is there in the slides itself. Okay. Okay, direct question. This is also purely 10th standard, I can say. 10th standard CBSE also you'll get this kind of questions. Which organelle in plant cell is responsible for maintaining turgor pressure and providing structural support? Obviously, vacuole is the organ which can act like a storage also, storage compartment, and most important, which plays a key role in maintaining hemostasis mechanisms. Maintaining what mechanisms? Hemostasis mechanisms inside the cell. So obviously vacuole. And ER involved in the storage also, SER, and RER is involved in the synthesis functions. You know it very well. And what about the function of the Golgi operators? Every student know that Golgi operators can act like a sorting station. What? Golgi operators can act like a sorting station. And you know, right, mitochondria involved in a multiple functions, majorly in the metabolical functions. Majorly. All right. Okay. Think properly and answer the question straightforward. This is also a 12th standard, NEET level question. 12th standard NEET level question. Okazaki fragments are formed during DNA synthesis. DNA synthesis extends from 5' to 3' direction only. Everyone. Again, simple, straightforward question. You all know very well, Okazaki fragments are formed during the DNA synthesis, especially in which strand? Lagging strand. Okay, and we have studied crystal clearly that whether it is a leading strand, whether it is a lagging strand. How many times I have told to all of you, the DNA synthesis always, the daughter strand synthesis always happen in 5' to 3' direction. We know it very well. Okay. I want you to answer this question. Okay, Padmavati, Vijay Lakshmi, Akila answer this question very correctly. We read it clearly. Especially whenever I am explaining you the DNA repair mechanisms, I have explained you. Which of the following statement is false about DNA polymerase 5 of E. coli? Swati, think and tell me. Tell me, Akila, think and tell me. This polymerase 5 is involved in which DNA repair mechanism? Vijay Lakshmi, Chandar, everyone is there when I am explaining you this. Padmavati, Shivani, where we read this DNA polymerase 5? No, I am asking a question to all of you. DNA polymerase 5, in which repair mechanisms that we have studied? That is my question to all of you. Everyone forgot, friends. I have to, very good. Correct, Akila. Translation synthesis or bypass mechanism, and even we also read in the SOS repair mechanism also. Very good, Selvi. Very good, Padmavati. It is in the last class only I told you, around 4 days back when we are doing the DNA repair mechanism, I have explained you this time a specialized mechanism called as translation bypass. We read it now. And remember my question very clearly for the exam. Among all the DNA polymerase enzymes, every student remember DNA polymerase 5 is said to be the most error prone enzyme. This is an important statement I want all the students has to remember for the exam. What is that point? It is more error prone enzyme. See friends, if it is already a error prone, how it can show the proofreading activity? It is not at all showing the proofreading activity. That is the only reason we called it as the error prone. I hope everyone is getting my points very correctly. DNA polymerase 5 is a specialized translation synthesis polymerase in E. coli. It belongs to the gamma family of the DNA polymerase enzymes. And what is the most important one? It is the most error prone because it lacks the proofreading ability. But they said that it has the proofreading activity, which is the wrong statement. Very good, very good. Okay. Now first, whenever I am explaining you the DNA replication in eukaryotes, I will always ask you this question. I don't know how many of you recalled this. I always ask you. Now tell me, Selvi, what is the enzyme first? Enzyme is present or not in the given options? Akila, Shivani, answer the question. First of all, tell me what is the name of the enzyme that is responsible for the mitochondrial DNA replication? Every time I use it to ask this question. Selvi, what is the question asked? Selvi, look into the question. They are asking which is false. Selvi, they are asking which is false. Which is false they are asking. Which is false. Okay. It catalyzes error prone replication. It is true option. No. They are asking the false option. Okay. Now, anyway, which of the following enzyme is responsible for mitochondrial DNA replication, which is called polymerase gamma? Now tell me, is this the symbol of gamma? I am not understanding actually. Is it a symbol that represents the gamma? I am not sure about it. Polymerase alpha, beta, delta. I think this will be the gamma, right? And this will be the epsilon. I think this is the polymerase lambda, no? Is this the gamma? No, right? No, this will be the delta. This is called delta. See, this is alpha, beta, delta, gamma, epsilon and lambda. Correct, na? This is lambda. But what is the question they have asked in the exam? Which of the following enzyme is responsible for mitochondrial DNA replication? But they have given answer as the polymerase lambda. So this is also completely wrong. It is completely wrong. So I am telling you sincerely, object this question. You will get the 4 marks. Slowly friends, you have to see the questions very carefully. Okay. I am sincerely requesting, aram se you will get the 4 marks. Just like that you will get the 4 marks. Okay. So this is completely wrong. It is the polymerase gamma. Okay. Compulsory, object this question. Easily you will get it. Because this is what gamma, but they have given this is the lambda, right? Yes. 4th option is wrong. This cannot be the answer. Please object it. Very good. Okay. Now, I want all the students has to give the correct and accurate answer for this question. Very simple, straightforward question. Correct. Lambda in the recombination. Correct Padmavati. Now, question is, Which of the statements are correct about the spliceosomes? Which of the following statements are correct about the spliceosomes? I don't know how many people can object. You can object lot and lot of questions. Which of the statements are correct about spliceosomes? Everyone, can any student please answer my question? Why the option 4 is wrong? Selvi, why the option 4 is wrong? Now my question is why option 4 is wrong? Akila, why option 4 is wrong I want. Why option 4 is wrong? Very good. Because you know very well option 4 is talking about what kind of a splicing? Option 4 is talking about the alternative splicing. Correct or not? In case of alternative splicing only, we skip the exon and we will rejoin with another exons. Of course, we read multiple times, spliceosome is the combination of RNA plus proteins, which we generally call it as the SNRPs. Correct na? Very good. Correct na? They recognize RNA sequences that signal for removal of introns, right? They can produce different mRNA molecules by splicing at alternate site. Okay. Actually, if the question is only about the spliceosomes, even option 3 is also wrong option. Because option 3 is also asking about the alternate splicing only. Even option 3 also. So if I can say most probably 1 and 2, but we can take even option 3 is also into the consideration. Okay, then option 4 will be the correct. No doubt about it. Correct, correct. Very good, very good, very good, everyone. Okay. Yes, okay. See, the common line that I use to teach you every single time in the class, the common line. The Shine-Dalgarno sequence is responsible for the common line which I use it to teach. So every student know very well this is the 5' to 3' mRNA and this is what sequence? AUG sequence. And exactly 10 nucleotide upstream to the AUG sequence, there is a complementary sequence is present. To what it is complementary? 30S ribosomal contains 60S ribosomal subunit and that 16S ribosomal subunit will come and attaches to this particular complementary sequence. Based on the name of the discoverer, we call it as Shine-Dalgarno sequence. So this is the binding site of ribosomes to the mRNA during the process of translation. Very good, very good. Okay. Now I am requesting every student has to answer this question. I don't leave any one of you if you do this question wrong. Especially I am telling to all the CSR students, you have to answer this question correctly. Anyway, the answer given is wrong. I want Swati, Padmavati answer it correctly, Selvi. You can object this question right away. I want every student has to answer this question. Take your time, no problem. How it is 450, Padmavati? Tell me. How it is 450? They are asking only length. No, no, no. Everything is wrong. I want everyone to choose it correctly. Okay, Afrin, tell me how. Everyone, Vijay Lakshmi, what is the answer? Use just a common sense. The question is crystal clear. The length of the final processed mRNA. I want you to remember this point. Length of final processed mRNA. You can object this question. Okay, no problem. At least give me the answers randomly. Whatever you think, whatever you consider. Okay, not a problem, not a problem. Let's continue, let's continue. Let's draw the mRNA. This is mRNA. Every time we use it to read, this is called 5 prime, 3 prime. Okay, Padmavati, understand here. Okay, you said is right. See that. This is called 5 prime UTR and this is called 3 prime UTR. How many times we all discussed all this? So 5 prime UTR, we generally call it as what? Untranslated region, also called as the leader region. Correct na? And 3 prime UTR, we also call it as what? Trailer region. Correct na? Crystal clear. Now, what is the question? They says that a gene encode a protein with 150 amino acid. So 150 amino acid means each amino acid is coded by 3 bases. So there are total 450 base pairs. No doubt about it. There is one intron of 1000 base pairs. Now my question to all of you is crystal clear. Whenever mRNA got completely processed, every student answer, is there any necessity to consider these 1000 base pairs? Every student answer my question. Whenever the mRNA get processed completely, is there any necessity to consider the 1000 base pairs of the intron? Everyone. Of course not. Of course not, we don't consider. So 450 base pairs, and what they said, the 5 prime UTR contains how many base pairs? 5 prime UTR contain 100 base pairs and 3 prime UTR contain how many base pairs? 200 base pairs. 200 base pairs. Now, shall we think properly and answer my question? Swati, think properly and answer my question. Once the mRNA get completely processed, think properly, don't be hurry. Once the mRNA get completely processed or matured, UTR will be present in the processed mRNA or not? My question is very clear to all of you. Whenever mRNA become completely matured, UTR will be present in the final matured mRNA or not? That's my question. Akila, does this particular UTR is present in the final and processed? How not? Everything you people forgot it. Swati forgot everything. Why will not give? Can you give me the reason? You are not at all understanding the points. Everyone use your brains. All of you understand the point correctly. What is called as RNA processing? What is called as RNA processing? RNA processing, we cut the intron and we add the exon. Now every student answer my question. Have you read anywhere that we are removing the UTR during the processing? Every student answer my question. Have you read that we removed the UTR during the mRNA processing? What we do during the mRNA processing? We add the cap, we do the polyadenylation, correct na? And we just do the splicing. Where did we cut the 5' UTR? and tell me what is the meaning of a UTR? Untranslated regions. Untranslated, which means what? They are traveling to the cytoplasm and they do not have any kind of a significance. UTR will be remain as it is. They do not have the significance. But what is the question they asked? What is the length of the final processed mRNA? So processed mRNA having 450 base pairs, 100 base pairs of the UTR, 550 plus here 200. So total will be 750 base pairs. This is the answer. Option 2. Okay. You can 200% you can object the given question. You can raise the objection. There are many questions that you can object. I believe everyone understand the question correctly. If they given the final processed mRNA 450, that is correct. If they did not mention anything about the 5' UTR and 3' UTR, they have very clearly given that 5' UTR contain 100, 3' contain 200. They will be present in the mRNA after the processing is over. Then the total length will be 750. Padmavati, is it clear? Everyone, Shivani, are you there? Swati, you understand? Vijay Lakshmi, Akila. Is it clear for everyone? Just let me know quickly. Okay. Anyway, I just kept the explanation. Look into this. 5' UTR contributes to the length of the mRNA and it is given 100 base pairs. 3' UTR, of course, it is not code for any protein, but the Akila question is super clear. They are asking only about the mRNA they are asking. They are not asking about the protein. The question is crystal clear. Final processed mRNA. Final processed mRNA. Okay. Answer is 750. Anyway, look into this. The 5' UTR contributes the length of mRNA given is 100 base pairs. 3' UTR also contributes the final mRNA length and is given as 200 base pairs. So to calculate the final processed mRNA length, we sum coding region, we sum 5' UTR, we sum 3' UTR. So total will be how much? 750 base pairs. Hence, option 2 will be the correct answer. You can compulsory object this question. Okay, no problem, no problem. Okay, so easy questions. Which statement is false about genetic control in prokaryotes? Which state? They are asking, friends, false. They are asking false. Everyone, we all said multiple times, suppose this is the bacteria. In the bacteria, the DNA replication is completed. When the DNA replication is completed, what is coming out? mRNA is coming out. Whenever the mRNA is coming out, what happened? Whenever a part of the mRNA comes out, immediately the ribosomes will come and attaches and start the synthesis of the protein. What is the meaning of that? In case of a prokaryotic organism, we studied very clearly that transcription and translation are coupled. Transcription and translations are highly coupled in nature. Correct, na? So, the gene expression majorly occur at the transcription level followed by the translation level. The gene regulation I am talking about. So, most of the gene expressions are regulated, understand, is translation level. So, which is a wrong statement. Most of the gene expression start at the transcription, occurs at the transcription, it completes at the translation level, right? Hence this statement will be a false option. No point of getting confused over there. All right, okay. Everyone think properly, use your brain, apply the knowledge and answer this question. This question linked, this question is a linkage between the virus and cancer. In the last time, whenever I am explaining you something called as a transduction in the genetics, that time also I have explained you this. Okay, just read it again. I believe all the students remember this. See friends, it is always idealized while solving the questions. Always remember my words, eliminate the options. When you try for finding the right answer, you can't guess the answer. When you try to eliminate the answers, eliminate the options, you can come to a conclusion. Now all the students try to understand my statements. Does a virus is involved in inhibiting the tumor suppressor genes? Never ever. Tumor suppressor genes either directly or indirectly cannot be inhibited by the virus. Correct na? Hence, option 3 is the wrong option. Correct na? You can eliminate the option 3 also, option 4 also. Then there are only two options are there. Now, you tell me why viruses are involved in the activation of the oncogene. Virus never involved in the activation of the oncogene. We all people commonly know that why virus cannot be involved in the activation of oncogene. What is oncogene first? Whenever a proto-oncogene, what is proto-oncogene? We read it crystal clearly. Proto-oncogene is a normal gene. Whenever it gets an extra function, what kind of mutation it is? Gain of function mutation. We read it, also called as dominant mutation. Whenever it gain its function, then the proto-oncogene converted into the oncogene. Then tell me, what is the role of the virus here? There is no role of the virus in activating the oncogene, or there is no role of the virus in inhibiting the tumor suppressor gene. Then you can eliminate the option 2 and 3 very easily. What is called insertional mutagenesis? Insertional mutagenesis means introduction of the viral genome into the host body. That is what we can call insertional mutagenesis. Look into this, everyone. Insertional mutagenesis refers to the integration of the viral DNA into the host genome. What it will do? Which can disrupt normal gene, potentially leading to the tumor transformation. So option 1 is absolutely true. Option 2 is wrong. Option 3 is also wrong. Direct inhibition of viral replication refers to the blocking the ability of the virus to multiply inside the cell. Correct na? Anyway, this mechanism is not involved in virus induced transformation. Of course, this is also true. But you all people has to understand what is the question they are asking here. Which of the following mechanism is not involved? They are asking not involved in the virus induced cell transformation, right? So option 1 and option 4. No point of getting confused, friends. Don't come to a confusion. First try to analyze the options very correctly. Some viruses will cause the cancer, there is no doubt about it. They are direct. I have mentioned that, Akila, you can see that HPV, human papilloma virus, Correct, Swati. It's like exactly like a bacteriophage. Swati, you can see that HPV inhibit the P53. Then it will cause it. But that is not the question that they are asking here. You're getting the point. They are not asking about that. They are asking a general characteristics that cannot be shown during the viral transformation. Okay, 1 second. 1 second. Let it load. Just 1 second. 1 second. That is true. Whatever you said is absolutely true, Padmavati. Look into this picture. All of you, whomever did not understand option 2 and option 3, look into this. Oncogenes are mutated or overexpressed versions of normal gene. I already told you there is a, no, Akila, look into this correctly. Look into this correctly. Oncogenes is what? Whenever a proto-oncogene undergo a dominant mutation, it is converted into oncogene. No doubt about it. Now everyone look into this. Certain viruses can introduce or activate oncogene in the host, leading to increased proliferation and potentially cancerous formation. Now you look into this. Many oncogenic viruses act through oncogene activation, making this mechanism involved in virus-induced transformation. This is also not the correct answer to the question. What is the question they are asking? Every student look into the question. No one is not looking into the question. They are asking the question, which of the following mechanism is not involved? Akila, whatever you said is right, but the question is opposite. The question is which is not involved. You all people said is absolutely right. The viruses can cause the cancer, but that is not the mechanism of the virus-induced transformation. The question is about transformation. Now you only think it. In the virus-induced transformation, why oncogene will enter? In the virus-induced transformation, what is the role of the tumor suppressor gene? These two are completely irrelevant to the given question. Then virus-induced transformation is what? Introducing the viral genome into the host genome. That's all. Right? Hence option 1 and option 4 are the more relevant options. They are asking which is not involved. Tumor suppressor gene and oncogenes are involved. I hope everyone is getting my point correctly without confusion. Look into the explanation again and ask me if you did not get it. I have written very elaboratively. All right. Again, straightforward question which is there in the slide itself. Which photoreceptor in plants primarily responsible for regulating the flowering time in response to day length? Which is directly there in the slide itself. This question. Still lot of questions are there. Please go ahead quickly. Please be patient. Don't leave in the middle. Which photoreceptor in plants is primarily responsible for regulating flowering time in response to day length? Of course, friends, phytochrome B. We already studied very clearly. Phytochrome A is primarily sensitive to the far red light. What I told you, if the phytochrome A want to show their function, the intensity of light should be very low. Correct, na? And if the phytochrome B want to show their function, I have explained it, Vijay Lakshmi. Once again, you watch the video. Phytochrome B wants to work effectively. It requires the high intensity of light. And the phytochrome B is the one that plays a key role in flowering in response to the day length. In day length, no point of confusion. This is the direct line which is there in the slides itself. So which is the phytochrome B. Don't get confused. So answer will be the option 2. Okay. See, this kind of questions, my friends, it is extremely common in the CSR net also. See, this is the question which is not there in our books. This is the question which is always you have to know, industrial applications, the recent trends. right? Suppose if you look into our CSR July paper, we got the question about the COVID vaccine, but you can easily answer the questions. Try it. These options also absolutely wrong options. There is no doubt about it. Okay, I'll tell you the answers. I encourage you to remember. This also you can 100% object this question. It is a generalized information. You can search in the Google also and you can get the information very clearly. Pfizer BioNTech. What this method is, they are generally using mRNA for delivery of the genetic material. Even Moderna, that is also name of the COVID vaccine, that is also using mRNA for their genetic material delivery. If you remember friends, you take an example of a Covaxin, you take an example of a Covishield also. Right, both what we are using, we generally using only mRNA. Correct na? The entire research is purely mRNA level. Same, this Pfizer BioNTech COVID vaccine and Moderna COVID vaccine, purely they utilizes mRNA for the delivery of genetic material. And you read it, and many complaints also has arisen, many disputes also arisen on the AstraZeneca, where AstraZeneca is using a virus. They used the viral vector. Even Johnson and Johnson, which is called Janssen vaccine, they also used the viral vector platform only, okay, for the delivery of the material. So this question also completely wrong option they have given. I encourage you compulsory object. So the correct answer option A and B will be option 2. Whereas option C and D, option 1. AstraZeneca and Johnson & Johnson, they used vector based platform, whereas Pfizer and Moderna used mRNA to deliver the genetic material. Simple. It is informative or fact based questions. It's not a fact based, informative based questions which you get the information directly. That's why it is much important. We have to be updated with respect to the recent trends that are happening in the life sciences or in the field of biotechnology. Right? This is also straightforward question. Everyone please answer. Tell me why. Which sequence of the following techniques in an antibody engineering involving the use of a transgenic mice engineered to produce human antibodies and subsequently immunized with the target antigen to generate fully human monoclonal antibodies is correct? I don't know how many of you are remembered this. Whenever I am explaining you what I can say, monoclonal antibodies, I have explained in the same slide to all of you. I don't know how many of you remembered during that time. Okay, 1 second, just give me one moment of time. I have explained you a special slide named as humanized monoclonal antibodies. Okay, just give me one moment. So this is the exact question they have asked. Okay, this is the question that they raised here. Okay, anyway, you try it. Yeah, everyone try it. Which sequence of the following techniques in an antibody engineering involving use of transgenic mice engineered to produce human antibodies? Friends, the question is very clear. They are asking the question about human antibodies. So what is the meaning of that? In the place of mice, majorly, we did the humanization. So I am giving you the hint. Obviously, humanization is the first method or the first step. It's very simple, friends. I believe you all people remember what is called as a hybridomo technology. I believe you remember we have taken a we have isolated, anyway I'll explain you here very quickly, try to understand what we have done here. I believe everyone remember, we have immunized the mice with a specific antigen, right? Then obviously the mouse body starts producing what? Monoclonal antibodies. Whether it is producing the monoclonal antibodies or not, we have rechecked with a blood sample. Once we confirm that the body is producing the antibodies, then what we have done? We have isolated the spleen cells. Why only we have taken the spleen cells? I have explained you this statement also. Spleen cells are more effective in production of the antibodies. So we have taken from the spleen, we have taken plasma cell, we have combined with the myeloma cell. Then what we produced? We produced the hybridomas. Then after production of this monoclonal antibodies, now you see that initially whatever the antibodies that we produced, which are completely isolated from 100% of the mouse protein. Later, as the time progresses, we used only 33% of the mouse protein, we used only 5 to 10% of the mouse protein, and we used now 100% of humanized antibodies. This is the evolution in the antibodies. Now they're asking what is the sequence over there. Sorry. Sorry. First will be the humanization, then hybridoma technology, fast display, combinatorial antibody libraries, which we generally call it as genomic antibody libraries. These are all the common steps that are involved in the human monoclonal antibody evolution. If you can remember that, then only you can answer that given question. All right, okay. Okay, so very straightforward question. I want every single student has to answer the given question correctly. What is the question? They are saying that, listen, every single question came from the slide. So developmental biology, plant physiology, human physiology, genetics, techniques. I want you to solve every question. Please be patient, don't leave in the middle. Which of the following is not a characteristics of GPCR? Friends, they are asking not. Once again, understand the given question. They are asking not. Of course, everyone. See, they are involved in transmitting the signal from extracellular ligand to the intracellular signaling pathways. How? Of course, the information can be passed with the help of secondary messengers. And we know that we call it them as the serpentine receptor because they can transpan the membrane 7 times. We read that. They possess an extracellular binding domain, transmembrane domain also. Right statement. They are exclusively found in the prokaryotic cells. That is a wrong statement. GPCR not exclusively found in the prokaryote. Actually, they are primary. They are primary receptors in case of a eukaryotes. There's no point of getting confused, friends. They're primary receptors in the eukaryotes, not in the prokaryotes. All right. This is also a straightforward question. I want every one of you to answer. What is the primary function of quorum sensing in the bacteria? Straightforward question which you read it already. What is the primary function of the quorum sensing in bacteria? See, quorum sensing is a mechanism where the bacterial cells are becoming adapted. Bacterial cells are becoming adapted to new changes in their gene according to the environmental influences, which is a straightforward direct Definition of the quorum sensing. Quorum sensing is a regulatory mechanism used by the bacteria to sense their population density through a chemical signaling called as autoinducers. Don't get confused with this. Now, because of a specific signal, they can form a biofilm. I believe you remember when the biofilm got formed around the bacterial cell, bacteria will become resistant to any kind of a stressful condition. So this kind of a mechanism generally called as the quorum sensing. Straightforward only. Okay. Now, every student answer the question. Which of the following best describes the function of the tumor suppressor gene? Everyone, please be patient. Please be at the end of the class. Correct. Which of the following best describes the function of a tumor suppressor gene? This is also directly in the what we can say that in the slides itself. You take any example of a tumor suppressor gene, you take the example of a P53. What the P53 tumor suppressor gene will do? First, it will arrest the cell cycle. Then once it arrests the cell cycle, it will activate the DNA repair mechanism. If DNA repair bioluminescence also correct. If the DNA repair mechanisms are failed, immediately what the P53 is going to do? P53 immediately release or stimulate the mechanism called as the apoptosis. Correct na? These three are the functions performed by a tumor suppressor gene. They repair damaged DNA to prevent mutation. Absolutely true. Okay. This is also correct. Now, option 3 is also correct. They inhibit a cell cycle progression and prevent a tumor formation. This statement is correct, no doubt about it. But what is the question they asked? Best describes. Best describes. This statement is true. I agree with everyone. I agree with everyone. This statement is true. But the best statement that describe is they activate the DNA repair mechanism to prevent the mutations. If we prevent the mutations, the cell cannot die. The cell never undergo the mechanism of apoptosis. See, every student see, option 3 is partially correct, but not the best answer here. While tumor suppressor genes do inhibit the cell cycle to prevent tumor formation, but their role in repairing DNA to avoid mutation is the broader role. You understand? That's the reason we can say that option 3 is the most, option 2 is the most appropriate option. Got it? Option 3 also true, I agree. Okay. This is also direct question. Which structural feature characterizes toll-like receptor? In the immunology, whenever I am explaining you the first class of immunology, we read about toll-like receptor, TLR, or in the second class of immunology. At least anyone remember? Selvi, you remember? We read TLR3, we read TLR4, we read TLR5, we read TLR9 receptor. Anyone remember? Which recognizes several pathogenic particles. Anyone remember this or not? Very good. So they are basically what? Extracellular ligand binding domain. Very good, very good. Absolutely true point, no doubt about it. Right. This is a straightforward question only. No confusion over there. Okay. See, I think I want every single student has to answer this. Answer Vijay Lakshmi very correctly. I have explained you just 4 days back this. You no need to look at the options also. We have explained just 4 days back this one. I don't want options also. You tell me the options. Just 4 days back I have explained this. More than 3-4 times I have explained you this. You no need of options also, you can choose the answer. Four or five times I have explained you this. Everyone, Akila, what is the answer? Shaila? Shaila, are you there? What happens? Some people did not attend. Just 4 days back we studied. Selvi, what is the answer? Swati? All are there in the slides directly. We know how many times I have explained. I don't know why people are not answering, okay? Bicoid, hunchback, nanos, and caudal. What is the function of these 4 genes? They are involved in the anterior posterior axis formation. So obviously D will be the one. How many times we have studied wingless gene, which is best segment polarity gene, correct or not? And hedgehog, also a segment polarity genes. Decapentalphagic, which is involved in the dorsal ventral patterning. Very straightforward question. I don't know why people are not answering this question. Same examples only we discussed in the exam also, in the classroom also. Very good, very good, very good, very good. Okay. This is for this batch we have not discussed this, but you can answer this. In the last batch, we have studied a concept called as apoptosis. And we studied pre-apoptotic protein and we studied pro-apoptotic proteins. Without option also you can give the answer. In the context of the programmed cell death, protein family contains members that promote cell survival by inhibiting apoptosis through binding and sequestering pro-apoptotic proteins. Arrange the following protein families involved in continue. Involved in programmed cell death in the correct sequence based on their roles. Based on their roles. Friends, you people can easily come to the conclusion. Actually, tumor suppressor gene will come into the picture very last. Look into this, every student. No point of getting confused over there. What is called as BCL2? I hope everyone remember this. Some members of this family are what? Anti-apoptotic proteins, which is called BCL2 itself, which promotes cell survival, while others are pro-apoptotic. Example, BAX and BAC, which promotes apoptosis. Right now also I am requesting every student remember the examples of pro-apoptotic and remember the examples of anti-apoptotic, compulsory for the exam point of view. Okay, pro-apoptotic, the major protein will be the BCL2, whereas anti-apoptotic BAX, you read multiple times, BAX and BAC. And remember, whenever you heard the name of a BCL2, it compulsory require the help of the mitochondria. Why? Because I have explained in the slide itself, BCL2 family always require another protein called as cytochrome C. When cytochrome C comes, then only caspases will be activated. Without cytochrome, caspases never will be activated. I believe you remember we read caspase 3, caspase 8, caspase 9. All together stimulate the apoptosis. How the apoptosis can be mediated? By the P53. And what is called tumor necrosis factor? Friends, I believe you all remember. Interleukins, TNF. What are all these, friends? These are all generally a cytokine molecules. These are all what? Cytokine molecules which are involved in the extrinsic pathway of the apoptosis. So the correct sequence will be number, what is the answer everyone has given? 3, 4, 2, 1. Everyone tell me. Others. So this is actually, this question also you can raise the objection. It is 4, 3, 1, 2. Why? Because friends, everyone remember one common statement. Very simple. Caspages. What is the function of caspages? Caspages are the one which will kill the cell finally. So remember, any time caspages are the one which comes last. Caspages. I told you multiple times, whether it is a BCL2, whether it is a cytochrome C, whether it is a P53, whether it is a tumor necrosis factor, everything ultimate goal is what? Ultimate goal is apoptosis. Destruct the targeted cell. The targeted cell can be destructed finally by an enzyme called as caspages. So undoubtedly caspages will comes later. At the end, see, use the brain, friends. First, what is required? First what is required? First required is the signal. Correct, na? So what will be the signal here? Undoubtedly tumor necrosis factor will be the signal. There's no doubt about it. Tumor necrosis factor is the signal. Then the hormone which stimulates the apoptosis. Then pro-apoptotic protein. Then finally caspase. I want you to compulsory remember, this is also a wrong options they have given. You can object the question undoubtedly. Okay, there are more than 12 questions you can object. Remember, it is all memory based question only. You have to remember compulsory. I kept everything elaboratively. I want you to read and answer the given question. Okay, this is also a question which we discussed exactly 4 days back. Exactly 4 days back we discussed. Exactly, I think last Friday only we discussed this. Vulva development in the C. elegans. We discussed in the last Friday or Thursday. Which of the following signaling pathways plays a central role in regulating the vulva development in C. elegans by coordinating sulfate specification and proliferation? I believe, friends, everyone remember, right? Notch delta signaling pathway. I believe every student remember what is the name of the signal molecule. Signal molecule is the Lin12. And what is the name of the receptor, which is called LECT23? And what is the signal molecule called as? EGF. Correct, na? And this is the receptor of the EGF. I hope everyone remembers. Straightforward question which we read it. It is there in the slide itself. Delta notch signaling pathway. Very good, very good, very good, very good. We studied it. Okay. Now this is also a question which we studied. I think someone asked the doubt also that time. I think Vijay Lakshmi is the one who asked the doubt. Okay. In the classroom, the same question was appeared in the exam. Same question. I think that time Akilo also asked the doubt about the limb bud. I hope all the students remember, this is the central axis. These are the limb buds. Correct, na? I hope you remember. This is also we discussed on Thursday or Friday class. This is the forelimb and this is the hind limb. Correct, na? And for the limb bud development, what is the major growth factor? I believe everyone remember FGF10. Correct or not? Fibroblast growth factor. Very good, very good. During the limb development in vertebrates, which signaling pathway plays a crucial role in establishing the proximal distal axis of the limb? Absolutely true. Fibroblast growth factor. Very, very good. Good, good. Perfect, perfect. We studied that. Okay. This I haven't explained, but in the last batch I have explained it. Arabidiosis Thaliana model. So easy model. You know, right, friends? The first oral will be sepals. You know, no? Others, first answer the question. Answer the question. Second oral will be the petals. Third oral will be stamen, fourth oral will be carpel. You all, you people remember this. Correct, correct. Which of the following floral organ identity genes is responsible for specifying the identity of a third oral in the ABC model of Arabidopsis thaliana? Straightforward question. So which is called as the AG, the name of the gene, which is called agamus. Every student listen, very simple question it is. The gene which can code for sepal and petal, we call it as aptella. Aptella 1, aptella 2. They are the one which is coding for the sepal and petal. Second and third oral, petal and stamen, aptella and pistilla. And third and fourth oral, stamen and carpels, agamus. So the correct answer is agamus. It's a straightforward fact-based question. Okay, if you just remember that model, you can answer the question very correctly. You know, whenever I start the concept of the gastrulation, the first line I'll use the same word. I don't know how many of you remember this. What is the first statement I use it to teach? Cleavage is the first phase of the development. I use it to tell you the same line. Gastrulation is the second phase of the development. Neurulation, you remember, which is called as final phase of the development. These are the common statement which we use it to read in general, is it? So embryonic stem cells are derived from which stage of the embryonic development? Undoubtedly, friends, it is from the blastocyst. Very good, very good. Okay, see, same question which we discussed again on the last Thursday. Same question we discussed on the last Thursday. In frog early development, what is the primary organizing center that initiates the formation of the body axis? Correct na? And which one is acting like an organizer? How many times we discussed? More than 5-6 times. Dorsal lip of the blastopore. Correct na? Dorsal lip of the blastopore. Organizer can be controlled or regulated by nucleus center. Henson node and primitive streak, these two are in the birds. Whereas in the frog, it is what? Obviously Spearman mangold organizer. Which can act like an organizer? Dorsal lip of the blastopore can act like an organizer. Organizer is regulated by the nucleus center. There is no point of getting confused over there. Whereas Henson's node and primitive streak exclusively of what we can say birds. Right. This is also a question when we started discussing about the fertilization topic in the developmental biology. This is also we discussed the same. Everyone, which cell surface molecule on the sperm is responsible for binding to the zona pellucida on the egg during fertilization? I believe every student remember this very correctly. Suppose this is the zona pellucida. Zona pellucida contain what? ZP1, ZP2 as well as the ZP3. Of course, ZP3 is involved in the stronger binding. And integrin, selectin, cadherin, what about these? These three are said to be what? CAM molecule. Cell adhesion molecule. They do not have any role in joining the sperm with the zona pellucida. Obviously, ZP3 is the right answer. Okay. Now, see, developmental biology, they have asked 8 questions. And 8 questions are easy questions. 8 are easy. Damn easy questions. Every question is just definition based, straightforward questions exclusively from the PPT level itself. Okay, that much easy questions that we can appear, we can expect in the set exams. Very straightforward. See, not even one logical, one experimental, any analytical question. No analytical question. Straightforward. Anyway, no problem. Now let's look into the plant physiology. The question says that please be patient, don't leave in the middle, we'll complete it 40 minutes. C4 and CAM pathways are more efficient than C3 pathway in minimizing transpiration. photo respiration. 32 marks, just like that we will get it. C4 plants spatially separate carbon fixation from the Calvin cycle, while CAM plants temporarily separate this process. Every student, what will be the answer? How many times we studied? C3 plant, C2 cycle occur. C4 CAM plants, C2 cycle never occur. So undoubtedly C4 and CAM plants, remember this is the question number 1. C4 and CAM plants are most effective compared to the C3. And I have crystal clearly told you, C4 shows the spatial separation. Why spatial? C4 contain mesophyll and bundle sheath. What about CAM? Temporal separation. Here it contain only mesophyll, but open during the night time, closed during the daytime. That's why we can call it as a temporal separation. Option 1 is absolutely right option. Okay. Don't leave. Please be patient. People do not have the patience only. Okay. And I am telling you very clearly, question number 92, you can object just like that. You can object undoubtedly this question. The all options they have given is wrong. Okay. Now all the students tell me, for the option A, what is the answer? For the option A, what is the answer? Every time we use it to read the same. Very, very easy questions. No, option, what is the option, Swati, here? Okay, symplastic loading, obviously wherever you heard the plasmodesmata, obviously option A will be 2, no doubt about it. But here they have given third option, which is absolutely wrong option, no doubt about it, absolutely wrong. Padmavati, don't forget here, A means what? It is the plasmodesmata, no? A will be 2, no point of confusing, okay? And this answer is wrong. 100% you can apply this. Correct na? And active unloading. Active unloading means what? It is an energy dependent process where the sugars are moving from the source into the sink. Again is the concentration gradient. So B will be the one. So actually option 2 is the correct option. B will be the one. And every student know very well what is called apoplastic loading, where the loading is occurring across the cell membrane. That is called apoplast. Passive loading means without energy. So D will be the 3, C will be the 4. No point of confusion at all. Okay. So option 2 will be the correct option. But in the question, they have given option 3. You can 100% object this. No doubt about it. No doubt. Okay. Next. Okay. We will do this question and we will stop it. Okay. Remaining we will do tomorrow. I think it's more than 2 hours. People are unable to sit. Which of the following statement about cryptochrome and phototropins are correct? Same question which is there in the slides itself. Which of the following statements about cryptochromes and phototropins is correct? We studied this, friends. I have, while explaining in the slides, I always use it to tell everyone, please remember the functions, please remember the functions, because functions are very important, friends. See, friends, it's very easy. We have 3 type of a photoreceptors. We have a phytochrome, we have a cryptochrome, and we have a phototropins. If every student recall, phytochromes, which are red light mediated, correct na? cryptochrome and phototropins. These two are blue light mediated. Very good, very good, very good. Mahesh Kumar, please be patient, okay? I'll do it, okay? Both cryptochromes and phototropin function as a blue light photoreceptors in plant, but cryptochromes are more involved in photomorphogenesis and phototropism, phototropins in light-induced gene expression. This is right. Don't join right now and don't ask me, okay? Right now. See, I am doing from starting. If you ask me middle, I won't do anything, okay? But anyway, for all of you, I am doing it very quickly. Okay. Everyone, Mahesh, just wait. I'll do that questions. I will give the PDF. You can see the objection. I kept crystal clear explanation also. Which of the following is a primary form of the nitrogen assimilation? Undoubtedly, you can object this question. No, this question. Right, this question is correct. This question is correct. We can't object this. Which if the following is a primary form of the nitrogen assimilation in plants? I hope you remember first NO2 we read, sorry, NO3 we reduced to NO2. Later NO2 we reduced to the ammonia. Right. So the primary form will be the nitrate reduction only, which is the straightforward statement, which is the right answer. Right. Nitrate reduction to the ammonia, no doubt about it. Mahesh Kumar, just wait, I'll solve your questions. I'm coming there. Which of the following is a common chemical defense mechanism used by the plants? Again is the pathogens. Very important question, friends, it is. See, activation of guard cells. Guard cells are actually involved in opening and closing of the stomata. Correct. Then production of cell wall degrading enzymes, no. There is no use of cellulase, pectinase. They never act like a defense mechanism. I will complete, Sandhya. Okay, don't worry. You people should be patient, will complete. And anyway, I kept every single question with explanation. You can see that. Okay. Synthesis of phytolexins. I want all the students to remember phytolexins are what kind of compounds? Antimicrobial compounds. Okay. So whenever the plant get any kind of a damage, we'll complete this. Immediately there is a release of this particular phytolexin. So obviously option 3 will be the correct. We'll do it. Okay. Question number 97. So this is also a direct question from the slide. I will give you the list of questions that you can object. Which of the following is a common adaptive response to a plant due to the heat stress? See, do not get confused here in the if they use the word water stress. In the given question, if they use the word water stress, then undoubtedly you have to choose the closure of the stomata. No doubt about it. But what is the question they asked? The question they asked is which of the following is an adaptive response to the heat stress? Whenever there is a heat stress, obviously the plant starts releasing certain osmoprotectants such as proline, such as betaine, such as mannitol. These are all can be released during the osmoprotection. So obviously what we can call the option 4, no doubt about it. Okay. Next, and some people are saying that this question is also wrong question. How this question will be wrong? Everyone please say that is this a true option or wrong option? Most of the people are saying that we can object this question also. You can't object this question. This is the correct question only, no? We cannot object this. See, we know, friends, P680, if there is a no presence of the pheophytin, obviously it is a plastoquinone, cytochrome B6F, then followed by the plastocyanin. So in the given question, they have not given the pheophytin. If they have not given the pheophytin, obviously after the P680, who will accept the electron? Plastoquinone only. Obviously answer 3. No doubt about it. Okay. Next question. When a person runs, very interesting question. It is very, very interesting question. When a person runs at a velocity of 100 meter per second and comes to rest after 30 minutes, then which of the following is the correct statement? The question is simple and use the brain. When a person runs at the velocity of 100 meter per second, comes to rest after 30 minutes. Everyone. See, obviously what will happen, friends? The person is running continuously and came to the rest. What generally happens? The body require more amount of the energy actually that particular time. Correct, na? Then very good, very good. Then glucose undergo the mechanism of oxidation into carbon dioxide. And but what about the water molecule? Water molecule generally released in the form of a byproduct. Always released in the form of byproduct. Don't get confused here. I just kept a very clear-cut explanation. When a person runs, the body needs to produce energy quickly to fuel the muscles. So what is the instant source of energy? Undoubtedly instant source of the energy is the breakdown of glucose only, which is the primary fuel. Correct, na? So you can see this picture. Whenever we break down the glucose, then it give rise to the carbon dioxide, and water can be released as a byproduct. You know, this is the common statement which we read from the 11th standard. Okay, now, and again straightforward question. This is also so these are all the questions from plant physiology. There are 7 questions appeared from plant physiology. Every single question is the most straightforward question. Most straightforward question. Any student can answer this kind of a question. Very straightforward. Easily. All right. Now next, look into the human physiology. Very straightforward. It is, 11th standard NCRT question it is. This is 11th standard NCRT question. Now, the maximum volume of air expired from the lungs by a person after taking a maximum deep inspiration. We read this, friends. Anyone can answer the question? See, it's a simple question. What you have to calculate? You have to generally calculate the vital capacity. What is the question they are asking? How much amount of the air that got exhaled they are asking. What they are asking? Exhaled. Exhaled after deep inhalation. So you have to calculate the vital capacity. In the classroom, I have clearly explained all the breathing patterns. More than 2 to 3 times I have explained the same thing. You have to calculate tidal volume. Tidal volume is fixed 500 ml. Inspiratory reserve volume around 3000 ml. And expiratory reserve volume around 1100 ml. Then it is correctly around 4600 ml to the 5000 ml of the air. No point of getting confused over there, right? Very good, very good. Answer is the 4. Straightforward question. Okay. And this question also you can answer it very easily, friends. Which statement is incorrect? They are asking incorrect. Incorrect. With reference to the structure of the mammalian heart. And tell me the reason why. I kept entire explanation very elaboratively. I want you to go through the explanations whenever you are objecting or whenever you are challenging a given question. Make sure that you see the question very correctly. All right. Okay. And the question says that which is incorrect. See, the interarticular septum has a small oval patch called fossa ovalis. Right statement. The right auriculoventricular aperture is guarded by a tricuspid valve and left atrioventricle by mitral valve or bicuspid valve. Very, very, very good, Vijay Lakshmi. Absolutely correct. Arioventricular valves are connected to the papillary muscles, not via bundle of the His. Very, very, very good point it is. Right. Option 4 is incorrect because auriculoventricular valves are connected to the papillary muscles through carda tendinae, not via bundle of His. This is the perfect answer, right? Very good, very, very good. It is the straightforward 11th standard NCRT based question. Okay, very straightforward. Okay, now look into the picture. Correct. Question number 1 or 2. The phenomena in which active transport and return of the molecules such as glucose, water, amino acids, calcium, sodium, potassium salts into the blood at the proximal convoluted tubule is known as. What is the question? See friends, the question is very clear. The question they says that return of the molecule. If you look into there are 3 steps are there. One is called glomerular filtration, one is called tubular reabsorption, one is called tubular secretion. If you remember my statements very correctly, in the tubular reabsorption, what happens? If any extra ions such as sodium, potassium, calcium, water molecule, essential amino acids, essential salts, if they are getting excreted, their excretion will be stopped and they will be reabsorbed back into the bloodstream. I have crystal clearly explained you with the help of a diagram, and that to depth we have studied in the class, which is called selective reabsorption. Why we are using the word selective reabsorption? That is also a very good question. Why we are calling selective reabsorption? Because these are all vital elements. Sodium, potassium, calcium, they are exclusively required to maintain the blood osmosis. That is the reason we should not allow these ions to go out of the body. So 100% we have to reabsorb them. That's why we use the word selective reabsorption. Okay. All right, Mahesh, please look into the 103 question that which you asked. Okay. Every student answer the question. According to the accepted concept of hormone action, if receptor molecules are removed from the target organ, then what will happen? Okay, Mahesh, please look into this picture. Okay, this is the plasma membrane. Let's consider this is a receptor which is located on the membrane. In the lessons, I have told you crystal clearly, if any hormone, any growth factor, any neurotransmitter, this is the first statement I use to explain to all of you. Any hormone, any growth factor, any neurotransmitter, if they want to show their function in the body or in the tissue, compulsory there is a requirement of the receptor. Compulsory there is a requirement of the receptor. Suppose example, Mahesh, let's consider you take an example of a glucagon hormone. What is the function of a glucagon hormone? The function of the glucagon hormone is breakdown of the glycogen into glucose. We know that very well. Suppose example, if the glucagon want to show their function, it has to attach us to the GPCR receptor. If the GPCR receptor I have removed it, how does the glucagon can show their action? They cannot show their action. Then there is a no production of the glucose. Obviously, yes or no? then there is an improper function of that specific gland or that specific organ. Option 4 is absolutely true option. The target organs will not respond to the hormone, because if the hormone want to show their action, receptor should be there. Now the receptor is not there, how does the organs will respond towards that hormone? Option A says that the target organs will continue to respond. That is impossible. Without a signal, without ligand, how can you expect the response? You cannot expect the response. This is wrong. The target organs will, it never respond. First of all, these three options are wrong. The correct, most appropriate option will be the option 4. No doubt about it. You can choose it correctly. And I have given you crystal clear explanation by considering each condition. The 4 conditions I have explained you can see that correctly. Because remember only one common point, Mahesh. When you remove the receptor, one point you have to understand, there is a no physiological response. That's all you have to remember. If the physiological response was not there, which means that that specific organ is not at all responding. That is the point I want you to understand. Option 4 is correct. Okay. You know, it is a general knowledge based question which we read in the developmental biology only. Which one of the following statements regarding test tube baby is correct? They are asking correct statement. Mahesh, not only the 4 questions you have asked, before I explained many questions, remember my words, before I have explained many questions, there more than 6 to 7 questions you can object 100%. By the end of the session, I will post the PDF in the Telegram, someone will post it, then please go through the questions that you can object, compulsory object it. More than 12 questions we can object. Alright. Which one of the following statements regarding test tube baby is correct? Fertilization of the egg is effected outside in the body, and the fertilized egg is then placed in the mother's uterus where gestation is completed. Correct na? Which we generally call it as what? In vitro fertilization. Correct na? So we already studied in a specific test tube where we add the sperm and egg. I have very clearly explained it to all of you. Some student asked the same doubt. Sir, how the sperm undergo the mechanism of the capsitation? Very good, valid doubt many people asked. How they will undergo the capsitation? Because without capsitation, sperm cannot fuse the egg. I told you very clearly how does the capsitation occur. Capsitation is possible because of the mild washing. Mild washing of the sperm head with the chemicals. I kept the very clear-cut explanation. I want all of you to look into this. Very good, GIFT method. Fertilization of the egg is done outside the body within a laboratory in a specific petri dish, which we generally called as in vitro fertilization. Obviously, option 1 is a right option. Most accurate description about in vitro fertilization. You don't need to think about other options also. They are very incorrect. All right. Okay. Okay. This is actually there in the NCERT only. Myopia. In the last Thursday, I believe all the students remember. Please answer me. While we are discussing the developmental biology, I have used one more word. You remember cyclopia. Can anyone tell me what is called cyclopia? The same statement we read it. The same statement we read it. Cyclopia, anyone remember? Cyclopia means what? There is only formation of one eye. You remember? And that one eye also occurs or formed at below nose level. I have very clearly told you this line. You know, more than 5 minutes we have discussed about this. Because of improper expression of the PAX6 gene. Anyone remember it or not? Eye develop below the nose because of the inhibition of the PAX6. Very good. Formation of the single eye. Whereas myopia means If you look into a eyeball having a specific diameter, then what happen? The eyeball will increase its diameter more than the normal. That condition only we can call myopia, which is a statement related question. Myopia also called as nearsightedness, a common refractive error in which eyeball is too long or the cornea is completely curved. Right, don't get confused. Now you can ask me, no, we can see the images, but what is the only disadvantage is that whatever the images that are located on the long distance, all the images will be blurry. So in a myopia condition, blurred vision occur. In a myopia condition, blurred vision occur. Simple, no point of confusion over there. Okay, similar kind of a question we did even in the yesterday class also. Not exact question, similar kind of a question we did in the yesterday class. Yesterday morning class. They are asking assertion and reason. You have to think properly. Use the brain. The question says that in a specific plant species, correct, it's dihybrid cross, two genes P and Q interact to determine flower color. The dominant allele is P, produces yellow pigment, while the recessive allele P produces no pigment. Dominant allele GQ can convert the yellow pigment produced by gene P into orange pigment. The recessive allele does not affect this pigment. A plant with a genotype PPQQ has orange flowers, and a plant with genotype PPQQ has white flowers. I want all the students has to remember this compulsory. All in a homozygous dominant condition, they are having orange flowers. Then all are in a recessive condition. Friends, all are in the recessive I am talking. All are in the recessive condition. They are having white flowers. All are dominant orange color. All are recessive white color. If they are in the heterozygous condition, are crossed, if two heterozygous plants are crossed, what is the expected phenotype ratio of the offspring? What is the expected phenotype ratio of the offspring? And now please look into the given options. When two heterozygous plants are crossed, the expected phenotypic ratio of the offspring will be 9:3:4. Next, the interaction between the P and Q genes result in the dominant QLL converting the yellow pigment produced by the dominant PLL into an orange pigment, while the recessive PLL produces no pigment. Everyone, straightforward question only, you can answer this. Very good, very good, very, very good, Vijay Lakshmi. Everyone. Others, please answer the question. All right, look into this. Both assertion and reason are true. All the students, please look into the given question, whomever did not understand this. The given assertion, when two heterozygous plants, PP, QQ and PP, QQ, correct na? What is the probability of getting all are orange? Obviously 9. What is the probability of getting yellow? Will be 3. The probability of getting all are white will be 4. Then obviously what? 9:3:4. So undoubtedly assertion will be true. Now all the students understand. Now if you look into the reason, this is more important. I want you people to understand. The interaction between the P and Q gene result in the dominant QLL converting the yellow pigment produced by the dominant PLL into an orange pigment, while the recessive PLL produces no pigment. What does it mean? This reason correctly explains the relationship between the P and Q. Friends, P gene produces what? Every student understand my words. P gene produces yellow pigment. I want all the students has to understand my statements. P gene produces yellow pigment. But what is the question? If the P gene wants to produce the yellow pigment, what P gene is required? P gene compulsory required the Q. I believe you have to remember this. Same thing I explained in the question. If P gene wants to produce the yellow, it compulsory require what? Q. Q. So Q gene to convert this yellow pigment into orange. So remember, if P is absent and Q is also absent, then what happen? You don't get orange, you don't get yellow. Then what do you get? You get the white. No point, no need to get confused. I will write it here once again. Every student try to understand. P gene originally give rise to the yellow, no doubt about it. If P gene in the presence of Q, it is converting yellow into orange, no doubt about it. So if P and Q both are absent, then what do we get? We get the white. That's what we are saying. Every student please look into this given question. Here P and Q are present. I believe all the students is getting my statements correctly. Here P is dominant, Q is also dominant. That's why I got the orange. Why? Because Q convert the P into orange. Now you see the second condition, only P is present. So only P is present, but the Q is in the recessive form. So it cannot give rise to the orange, it can give rise to the yellow. And P is absent, Q is also absent. Both are in the recessive form. Then it is going to give rise to the white. Hence both reason and assertion are absolutely true. Is it clear everyone? This question, is there any confusion? One second, it will load it. Is it clear everyone? Any confusion? I will re-explain, let me know. Is it clear? So I'll tell in a simple words. P only gives yellow. P in the presence of Q, yellow converted into orange. If P and Q are absent, we get the white. Simple. So when two heterozygous plants are crossed, the expected phenotypic ratio is 9 orange, 3 yellow, 4 white. Assertion is true and explanation is also absolutely true. The interaction between the P and Q genes result in the dominant QLL converting the yellow pigment produced by the dominant PLL into an orange. Absolutely right. No doubt about it. Very good, very good. Okay. If you know the mapping formula, then only you can answer this question. But I believe, anyway, look into this. Everyone try to answer this question, which we are going to discuss in the, either tomorrow or in the Monday class. That is linkage and mapping. They have asked the question in the tetrad analysis. Anyone, if you can try it, give a try. Not a problem. Again, I'll explain in the next class. In a tetrad analysis of the Neurospora crassa, there are two linked genes are there. One is A gene and another one is the B gene. And they have given the following tetrad. Parental ditype 80, we call PD 80. Non-parental ditype NPD 10. Tetratype TT 30. Without knowing the concept, you cannot answer this question. But I'll tell you the formula, remember. Given these observations, what is the question? What is the distance between the A gene and B gene? So there is a simple formula to calculate the map distance. Every student just try to understand how you have to calculate the, one second, let it load. Yeah. So see this, how can you calculate the map distance? Number of tetratype plus 0.5 into, what we can say, non-parental ditype by total tetrads into 100. So here everything they have given, everything they have given. You have to just remember the formula to calculate this particular map distance. They have given PD type, they have given NPD type, they have given TT. Just apply the formula. Obviously you can get the answer of the 29.17 map units, which is approximately 30 map units. Here nothing to explain, friends. You have to remember the given formula. In the question they have given PD also, NPD also, TT also, and they have asked us distance. In order to calculate the distance, first you have to understand total tetrads. Total tetrads can be calculated by parental ditype, non-parental ditype and tetra type. Then you will get the total tetrads. So then just substitute the total tetrads in the given formula. Obviously you can get the answer. In the next class, either tomorrow or Monday class, I'll explain the linkage and mapping. See, this is the same question and this is the same slide. Same question, same slide we read in our lesson. Okay, I'll show you the same slide also. Okay, what is that? Anyone remember this? Bacterial genetics. Same slide only we read it. Just a moment, I'll show you just. I'll just show you one, just a minute. Yeah, can you see this? This is the same slide they have given in the exam. Okay, 5 minutes, 10 minutes, 15, 20, 25, 30. You know, the same type of genes that whatever we have studied in the same slide itself. Now, what is the question? In an interrupted mating experiment with E. coli of HFR strain. This is one HFR strain and this is one F minus strain. All the students, please look into every student, please answer my question first of all. I want all the students has to answer. I believe everyone remember. What is the resultant copies that we get whenever there is a mating occur between HFR and F minus? Padmavati, tell me. Vijay Lakshmi, tell me what is the resultant. Akila, please tell me what are the resultant copies. Chandar, you are there, na? What happens whenever there is a crossing occur between HFR and F minus? I believe at least one person remember. Whenever there is a crossing occur between HFR and F minus, actually there is a no change. What is the resultant copy? Resultant copy also HFR and F minus only. But what I told to all of you, what is the formula I told you here? What will happen? I told you very clearly to all of you, auxotropic strain converted into prototropic strain. This is what we studied. Whenever there is a crossing occurred between the HFR and F minus, very, very good. There will be a no change. Correct, Selvi? We can use the same. This will be same, but oxo converted into the proto. Now, can you see that what is the F minus strain? It is histidine, leucine, threonine, lac, galactose. And here all are positive. So obviously what is the order of gene? Order of the gene will be leucine, threonine, lactose, galactose, histidine. Simple. The option will be the number 1. Simple point. Just auxotropic strain converted into the prototropic strain. Very simple, straightforward question. No point of getting confused. Okay, there is one more question asked from the linkage and mapping, but this is a very straightforward question. Without doing the calculation also you can answer this question, because you have to remember the values. If you remember the values, you can easily answer it. Everyone try this question. Anyone can try. See friends, if you can remember the values of the linkages, you can easily answer the question. You have to remember negative LOD score, you have to remember positive LOD score, and you have to remember significant linkage. If you remember this, you can easily answer the given question. Anyone remember? Minus 2.1 means, now you people only answer. I will tell you, you people answer. Minus 2.1 means there will be a no linkage. 1.5 means there is a moderate linkage value. Correct na? Then what we can call the 2.7 indicate significant linkage. Significant linkage. 1.2 which indicate weaker linkage. These are all the standard values. 0.5 indicates no linkage. That's all. You have to remember this. If you remember this, you can answer the given question. Now tell me, what is the answer? See, theta equal to how much they have given? 0.2. Okay. Now you see this. Theta equal to 0.1 they have given. 0.1, what I told you, it will be the moderate linkage. So option 1 is the wrong option. Theta value is 0.2. 0.2 means LOD value is the 2.7. 2.7 means what? Significant linkage. That is the right option. And theta 0.2 means no significant linkage, wrong. 0.4 means what? There is a no linkage only. But they said that there is a no significant linkage. Answer is option 2. What you have to remember for that? You have to remember only the constant LOD scores. If you remember the constant LOD score, easy to remember. I'll tell you friends, just only remember this. If LOD score is 3, you always remember it is called significant linkage. Where LOD score is of -2 or lower suggests that no significant linkage. Very, very simple. So you have to remember negative linkage, positive linkage, and you have to remember the values. And in the given question, they asked only theta equal to 0.2. So LOD score will be 2.7. The linkage is significant. No point of getting confused over there. Straightforward question, which you can answer if you can remember the given values. Simple. Direct question, which is there in the slides of structural and numerical alteration concept we have studied. In the structural and numerical alteration, there is a direct question. It is, which type of chromosomal translocation involves the exchange of genetic material between non-homologous chromosome without any net gain or loss of genetic material? I believe you all people studied. One is called reciprocal translocation and another one is called as what? Non-reciprocal translocation. We read it very clearly. Reciprocal means what I told you, there is a two-way exchange. Correct na? Non-reciprocal means what? Only one way of exchange. How much ever I am giving, I am expecting the same. Hence there is a no gain or there is a no loss. Undoubtedly the answer will be the reciprocal translocation. There is no point of getting confused. Answer is 2. Anyway, I kept crystal clear explanation. You can go through it. Okay. And this is also a question which we have studied in the last time. In a study of non-homologous recombination in human cells, researchers observed that there is a breakage in the double strand. That double strand break we repaired by joining DNA ends with a little to no sequence homology. And what do we call that particular repair? Non-homologous end joining. Any students remember what are the name of the proteins that we have studied here? At least one fellow remember. I told you KU70 or KU80. Remember anyone? At least one fellow. And ligase. PKCS enzyme, which are just joining the ends. You remember KU70 and KU80 will associate with here and they're just joining that particular ends. Remember anyone, at least one fellow? Non-homologous end joining. We discussed in the DNA repair mechanism. Straightforward question it is. Very good. I believe at least someone remembered it. Good. Same thing. Okay. Okay. See, this is the question which we discussed in the previous Monday. Okay, previous Monday. Okay, when we are discussing a concept called as extra chromosomal inheritance, we have started reading a concept called as maternal inheritance. The same lines we all people studied. Tell me, Akila, what is the answer? We crystal clearly studied that there are three possible conditions are there. One condition, female is white, male is normal. And what about the offspring? All the offsprings are white. Next condition, female is normal, white, male is white or green, let's consider anything, doesn't matter. So all the offsprings are also normal. If the female is variegated and the male is normal, and what about all the offsprings? All the offsprings are also variegated. I don't know why others are not answering. Chandar, you are also there, na? Forgot it. Very good. Mirabilis Jalapa. Very, very good. We read the same. I explained 1 week back only, no? In a cross between variegated leaf plant. You remember what is called variegated leaf? It is little bit green and little bit white. Correct, na? I'll quickly show you. Very good. Which is called as what? Uniparental inheritance, and which we generally call it as what? Maternal inheritance. I believe every one of you remember that very clearly. Yes, this is the exact question, right? They have asked this one. So if it is a variegated, obviously the offspring is also a variegated, because offspring contain, mother contain both leucoplast and chloroplast. No point of getting confused over there, and it is easy to remember also. Correct. Very good. Very good. Very good. All right. Now this is also simple question. You people try and answer it. Try this, Swati. If in a study of a height in a particular human population, the heritability h square. This is actually a formula, friends. You have to know this formula. If you don't know this formula, you cannot answer this question. Just try to remember these formulas actually. Okay. In a study of height, if you are not confident about this, you can leave this, because there are many number of questions to answer. In a study of height in a particular human population, the heritability of height was found to be 0.8. Which of the following statements is most accurate? First, you always try to understand what they are asking. No problem. You have to just remember the formula. That's all. First, understand the given question. Understand. The heritability of a trait denoted as h square. First, every student has to understand what is h square. What it is mean? It means what is the total variation in a trait in a population. I am repeating my statement. First of all the students has to understand what is called h square. H square is generally explaining us what is the variation of a specific trait in a given population. Now you tell me, what is the trait? First of all, what is the trait? The trait here given is the height. Correct, na? The trait given is the height, which is 0.8 or 80%. Simple. So listen, heritability refers to the proportion of observed variation in a trait that can be attributed to genetic variation among individuals in a population. A heritability of 0.8 means that, understand everyone, 80% of the variation in the height in the population is due to genetic differences, while remaining 20% is due to environmental factors. It is the question that we can answer, by understanding the common sense, which is obviously this one. Because friends, here you have to consider the percentages. So they have given one trait is 0.8, correct na? Which means that 80% of the variation in the height due to genetic and remaining 20% because of the environmental factors. Simple. That is called as the H square. Okay. Firdos, I'll explain you. Let me complete. There are some 20 questions only there. We'll complete this and we'll discuss the cutoff. And this is the 11th standard question. Again, they have given. Every student answer this. Of course, they are the unicellular organisms with a prokaryotic cells. Okay, very simple, 10th standard question. I want compulsory, everyone has to answer this. The question they are asking is, which of the following statement is not accurate? Friends, they are asking not accurate. Yeah, you have to read again. Not. Everyone, come on, please be patient. 2 hours. See, the question is that not accurate. Can you see the option 2? Option 2 says that each organ in a plant or animal body is composed of a single type of tissue to perform a specific function. You all know very well, a group of cells give rise to the tissue. Correct na? Now you know very well, group of different tissues can give rise to the organs. Correct na? Hence, each organ in a plant or animal body is cannot composed by a single type of tissue. Please don't get confused with that. Okay. Next, again straightforward question which is just a definition only about phylogeny or evolutionary tree. Everyone, we all people commonly know that what is called phylogeny. Phylogenetic relationship, it is a definition at the 11th standard. It is an evolutionary relationship. Correct na? We are basically studying a evolutionary relationship among the different groups of organisms. That is what exactly we can call the phylogenetic tree. Very straightforward question. Next, which of the following correctly identifies the major habitat types found in the subcontinent of India? See, it's a straightforward question, friends. They are asking subcontinent of India. Tropical rainforest, deciduous forest, deserts, and mangroves. Correct or not? Don't get confused. It's a very straightforward question only. We can easily answer that question, right? Simple. Okay. One second. Yeah. Okay. Everyone try this question correctly. Question number 119. Which of the following statements about the intestinal parasites are incorrect? Friends, they're asking incorrect. Incorrect. They're asking incorrect. Anyway, it's also no NCRT question only, very simple questions. See, pinworms are not a flatworms. Pinworms are what? They are generally a roundworms. Correct na? Best, they are all belongs to the category or the phylum of nematoda. Don't get confused. So this is the only statement which is incorrect. Okay. Anyway, is it clear everyone so far? And remaining are also very simple questions. Then now I will tell you the cutoff. Okay. Is it clear everyone so far? Remaining all are very easy. 20 questions are left out. I'll give you this. You people can read this. Clear everyone? All right. Okay. I am just giving you the expected cutoff. Okay. I am just giving you the expected cutoff with respect to the all categories. I am just saying, friends, expected. Okay. Expected. Okay. It will be 52, 52, 52, 52. Okay. And I am writing here, and I will upload the document in the Telegram. Someone will upload BCA, BCB, BC. Yeah, you people can leave it. BCD, BCE, ST, SC, sorry, SC, ST, EWS. Okay, I am just giving you the general cutoffs, friends. It may be varied. Okay, I am just giving you. Okay, it may be 47 to 49. Okay, and BCB also, it is a 48 to 50, and BCC it is a 46 to 48. BCD again, it may go up to 48 to 50. BCE it will go 48 to 49. SC it may go up to 46. ST it may go up to 46 to 47, and EWS it may go up to 45. Okay, these are all the expected cutoffs that I am giving. It may be range 1 or 2% little bit different. All right. Okay, that's all for now. I'll please go through all the questions. There are 20 questions are left out. I am requesting you, please solve those questions also. It just take 10 minutes for all of you. All right. Okay, thank you all of you. Thank you.